good morning, wherever you are in the world. Welcome to this webinar with Merck and BioMedics about our call for application on new strategies to enhance the immunogenicity of tumors. My name is Rita Freischlad. I'm the BioMedics Events Manager, and I'm based at our beautiful institute in Heidelberg, Germany, which you can see behind me and Thomas. Speaking today are Thomas Röckle from BioMedics and Wei Shaw from Merck. And also in attendance, we have Sven Lindemann and Julia Meyer-Huberton. So Thomas, would you like to quickly introduce yourself?
Yeah, thank you very much, Rita. Hello, good morning, good afternoon, good evening, or simply good day, wherever you are in the world. My name is Thomas Röckle. I'm the head of research here at BioMedics Institute, and as Rita mentioned, in the beautiful Heidelberg area.
Wei.
Hello, everybody. So my name is Wei, Associate Director, Merck RU Oncology, and I will introduce myself in more detail later.
Thank you. Sven?
Yeah, hi, my name is Sven. I'm working at Merck. It's a beautiful location of Darmstadt, and I'm a Senior Innovation Manager, and concerning BioMedics, my active role is a Key Account Manager.
Thank you. And Julia?
Yeah, hi everyone. My name is Julia Meyer-Huberton. I'm a principal scientist in RU Oncology as well, working together with Wei on this new BioMedics program and being there for you in the Q&A, everything regarding the DDR questions.
Thank you very much. Thank you all for being here. Before we start, I just have a few brief housekeeping remarks. So if you check your toolbar in the Zoom window, you should see a Q&A box. We ask that you enter all of your questions using the Q&A box, not the chat. You can ask your questions throughout the talks, but we will get to them during the Q&A portion of the webinar. You can also upvote and like other people's questions. In a moment, I will share more information on the call, including some helpful links. We're recording this webinar, and I will send the link to all of you within a few days, and we will also be posting it on our BioMedics YouTube channel. And finally, after the webinar, if you have any further questions on the application process, you can email my wonderful colleague and our communications and recruiting manager, Bianca Christian at fbc@bio.mx, and I will share that as well in the chat shortly. So let's get started. Thomas, would you kick it off?
My presentation? Yes, I'm going to share. Hold on a second. Good. Do we see my presentation?
Yes.
So yeah, thanks Rita for the kind introduction. I'm here on behalf of BioMedics, and I'm replacing actually the founder and managing director of BioMedics, Christian Tidono, who had an eye surgery last week, and eyes needed to do a webinar. So I'm replacing here today, and I'm very happy to give you guys a feel for what BioMedics is all about. And then later you're going to hear from Wei what this call is all about. So before we go into BioMedics, just want to briefly introduce myself, that you know to whom you speak later. So my career journey is really between academia and industry, which is in the heart and the essence of BioMedics. Academia, obviously, got a PhD in Germany, University of Ulm, did 2 postdoc fellowships, one in Switzerland, one in the United States. And then I joined industry in 1999 and worked there for quite some time at Serono and later on at Merck. On the Geneva side, was involved in medicinal chemistry, drug discovery in various therapeutic areas. In 2012, I joined a non-for-profit organization as a director in translational medicine, where I developed medicines for malaria to treat malaria in children in Africa and other endemic countries. And in 2017, I joined the clinical stage biotech company as the chief development officer, working on monoclonal antibodies and again, clinical trial application. And in 2020, I joined BioMedics Institute as the head of research, and I'm representing BioMedics today in this call. So BioMedics is, we really seed biomedical innovation. What does this mean? Well, let me start first by saying that we actually celebrating this year our 10 years anniversary. We are an independent biomedical research institute, and we are embedded in the campus of the University of Heidelberg. I'm going to show you in more detail how this looks like. And we really combine the best of both worlds, academia on one side and the industry mind on the other side. And this unique innovation model that was developed at the time, together with Christian Tidone and Ulrich Betz from Merck, was published in Nature Biotechnology in 2015, which combines global crowdsourcing and local incubation. What do we mean by that? So BioMedics receives challenges from our pharma partners, which we then send out to the world, to you guys, and we do an actual call for application. So we ask you, how would you tackle actually that challenge? And then we get back solutions, ideas, and the talents in form of you guys. And we select carefully then the best proposals and the best candidates to execute on the project. The way we do that is actually in a three-step process. Number one, as I mentioned, the global crowdsourcing. So we ask the world, how would you actually tackle the problem? And we get in typically 100 to 200 project proposals from all around the world, up to 80 countries per call. We then would thoroughly select the 15 best proposals with the candidates and invite those 15 people for a 5-day boot camp to BioMedics, to an innovation boot camp, where interdisciplinary team building and mentorship will take place. And at that stage, the outstanding ideas will then be transformed into a well-designed project. There will be one winning group leader candidate out of that innovation bootcamp, which we then would relocate to Heidelberg together with spouse, and would organize kindergarten schools for children, find homes. And the winning group leader has the possibility then to build a team of up to 5 researchers and executes the project according to the deliverables. He will not do this in isolation because there's a significant amount of mentorship from, in this case, Wei and Julia, from myself, and also from an academic mentor. And what is very important, not only executing on the project towards the deliverables, what excites the pharma partner, but also the opportunity to publish. This is a typical photo of a bootcamp that we conducted in 2021 together with Merck. And you see actually photos of people on the screen. We did this in a hybrid format. So pandemic did not stop us from conducting those bootcamps. And you see here, the gentleman here in the front, this is Alex. He's now the group leader for Team EDC. And actually he chose this postdoc woman here who was on remote, is now on his team. Let me move away this bar because I don't see my titles, which is so. There's many things we really embrace, and many things that we actually allow our fellows, because we want them to be as creative as possible. But there's 3 things we should not change, and the 3 things we would like our fellows to really adhere to, and this is our core values. Now it's not going up anymore. It's blocked. What's going on? Now it's coming. Sorry for that. So yes, number 1, cross-leverage. We really inspire and motivate each other to unleash our full potential. What does this mean? We don't work in isolation. Currently, we have 10, 11 groups, and they work partly together. They inspire others. We really work under the concept, we grow if we help others. We don't hide our results. We look into how can we help our folks, our fellows to improve themselves. We cross-pollinate. We embrace diversity, not only from a gender, culture, or ethnic group, but also simply from the science perspective, bringing together various disciplines, because we believe the more people look at the problem from the outside, from various angles, the more innovation is happening. And obviously, the third one, it's not a mediocre work that we would like to achieve, it's aim high. Our goal is to enable a new generation of targeted therapies. So we're really aiming high in our work. We've been doing this, as I mentioned, for about 10 years now, and you see here a list of 6 major pharmaceutical companies with whom we have collaborated. We have a good track record of trust and performance with these. 21 research groups we started in Heidelberg so far. 10 of those were successfully completed, means 11 are still ongoing. The IP was then transferred, IP stands for intellectual property, was transferred to the pharma partners. So the pharma partner said, this is sufficiently interesting for us, so we're going to acquire the whole knowledge that you guys generated. And we really have expertise in oncology, immunology, and neuroscience, so a broad range of therapeutic areas. Our R&D focus in Heidelberg and the expertise really lies in human organotypic disease models. We have several teams that work with whole human organs, develop organ-on-a-chip models, for example, intestinal epithelium, synovium in RA, or the lung epithelium in COPD. Then combining these complex 3D models with multi-omics data, and not only the data, but also machine learning, and what nowadays is becoming very trendy is artificial intelligence tools to really make sense out of the data. And here we have two examples, T-cell dysfunction in inflammaging, which is one project that is sponsored by Merck, and another one, RNA splicing in cancer, which was also a Merck project that's recently completed. And also biosensors we're developing, which is certainly at the outer side of overlap with regard to the previous two points I mentioned. We had a project once where graphene-based nanosensors were developed, and currently we have a project very challenging where a team is developing a fiber optic sensor to detect the molecules in the brain of living animals. So biomedics is in the center, in the heart of a triangle of three rivers. The Rhine, the Main, and the Neckar, which runs through Heidelberg, really in the center, where about 100 startup companies and small companies with about 4,000 jobs in R&D are located. In this triangle of about 100 kilometers, you'll find 7 global pharmaceutical companies with about 50,000 jobs. 9 universities and research institutes are between Wiesbaden, Karlsruhe, Frankfurt, Darmstadt, Mannheim, and Heidelberg, as well as Ludwigshafen. And if we zoom further into Heidelberg, so on the northern part of northern side of the river, you would find the so-called Neuenheimer Feld. Feld stands for field, which used to be actually a potato field decades ago, and now is one of the most vibrating science centers in Germany. So on this square mile, you will find 100 institutes, 20,000 jobs, and about 16,000 students in biotech and in medicine. And within that, there's the technology park with about 40,000 square meters of lab and office spaces for companies like us. Further zooming in, Biomedics Institute currently has around about 2,000 square meters of open innovation lab in an office sharing community. And as I mentioned, we are on the technology park of the University of Heidelberg. Any technology that we won't have on site, we would get from our prime partners in close proximity. EMBO, just up on the hill, the University of Heidelberg, and the DKFZ, which is just across the street for us. These are major institutions who hold basically all technologies that one can imagine to conduct breakthrough science in medicine and in biology. So we actually have framework agreements with these organizations, which would make it very easy for you guys if you need, for example, a highly sophisticated electron microscopy technology. You could easily go and measure at the University of Heidelberg. It would not have to go through a long, lengthy contracting procedure. On top of that, our close friends and neighbors, we have around about 60 collaborations around the world with a lot of great institutes, framework agreements where all the logistics and the paperwork is done, which allows us to get cell lines within a week, to get mice within weeks, which to get very specific plasmids in days. On top of the Heidelberg, which has been a successful story for about 10 years now, about 2 years ago, Christian Tidona, our founder, had the idea to expand our model from research teams to more startup companies. And this in a very contemporary field, artificial intelligence. It's called the Ion Labs, and it's based in Israel. And it's a consortium of several pharmaceutical companies, as you can see here: Merck, Pfizer, Teva, AstraZeneca, and the Israel Biotech Fund is also supporting it, and AWS. And it's all supported by the Israel Innovation Authority, and the Weizmann Institute of Science is a strategic partner. And actually our location is just next to the Weizmann Institute in Israel. So there in Israel, we're not forming research groups. Actually, we're building independent startup companies in the field of artificial intelligence. The focus is truly on artificial intelligence, as I mentioned, and not so much exploring deep biology as we do here in Heidelberg. And multiple sponsors are on the project, whereas here in Heidelberg, on this call, as we discussed today, we have a single sponsor. And here the 5-year objective is actually to seed and incubate 20 startup companies. So you see, this is a second pillar of the whole biomedics family that we've been building over the past 2 years, and it's actually ongoing with boot camps there in Israel. And there's a third pillar coming up now with a call that is already closed in the boot camp to start in May, and that's on the US ground at Yale University. We will have our first biomedics project there in beautiful Yale University, and you're going to hear more about that if you follow us on social media. So activities at biomedics, what do? So we engage. We have special interest groups in lab tools, methods. Some of folks are dedicated really to show what's the brand new, what's the latest technology. Others focus more on how can we develop ourselves best, career development. What's our next step? What do we need actually to join industry? What do we need to become a professor? Science. We do every Friday meetings where one of our folks need to present in front of the whole audience in a comprehensive manner that the cancer research scientist understands a neuroscientist project, or a sensor technology understands the physics of another project. We do an annual retreat, once a year, annual, where we meet like in a scientific meeting and congress. Education, we offer a scientist entrepreneur course for those who want to become independent. Scientific writing. Also, in Heidelberg, you can easily survive with English, but if you speak some German, you probably get the right britzel at the bakery, so German classes we offer as well. At the campus, we have lunch talks together with the DKF Stat, where folks can invite professors, give a talk. And obviously, physical exercise is important, so we do a lot of sports activity, contribute to the half marathon, Heidelberg Man, dragon boat racing, climbing, yoga, you name it. It's a never-growing list. And socially, as many other institutions as well, potluck, having beer hours, hanging out together. It must be fun, it should be fun working together. And this is what we really embrace, also by an annual summer party, as we do this year for the 10th anniversary, year-end cooking event where we all cook together and have to eat our own food. And really the scientific retreat is a really an event where we get very close together, because we discuss, as you see in the right picture, science like if on a scientific congress, but also we celebrate in the evenings, do karaoke. And if you imagine, currently we have about 70 staff at Biomedics with more than 25 different countries, 25 different languages who sing karaoke until 3 o'clock in the morning is really fun. You see a picture here of the dragon boat race. And last year, this were the Biomedics folks were the Smurfs. We also, and a bit more seriously, we also have in all our teams one research assistant, as we call, who starts a PhD program with us and finishes their PhD thesis. So you see here a fellow from last year who defended her PhD thesis. I also mentioned that we do cooking classes, and we celebrate together as it is wished. Now, what's the value proposition for talents like you guys? It's fully funded project for 3 to 5 years. It really focuses on outstanding research and publications. It's a creative cross-cultural teamwork atmosphere. So a lot of different nationalities, a lot of different backgrounds, a lot of different angles that would enlighten your thinking. You get guidance from experienced mentors, like the industry mentor, Wei and Julia, you will hear from them later, like myself and a few others who have extensive experience from academia and industry in guiding you in the right way in your project. And you will be in an outstanding environment with direct access to leading research institutions and companies. When you walk through Heidelberg, you breathe and you feel the science. Leadership and entrepreneurship training. If you want to go independent eventually, we do courses on that as well. And it is a risk-free assessment of possible future career in a startup, a big company. Because many people don't know, am I ready for industry? What is industry actually all about? Do you want to pursue an academic career? Try out biomedics for a couple of years. You combine both, best of both worlds, and you'll see. Where do we recruit our folks from? From major institutions around the world. Here's a selected list of institutes and universities. And where do actually our folks go afterwards? And it's really 50-50. 50% of our folks then go to industry. You see familiar names here of major pharmaceutical companies, but also smaller companies. Or some say, well, actually, I love academia. So I continue my career, become a professor, become a lecturer, or become a senior postdoc if this was a postdoc position at BioMedics. And with that, I'm actually done with my presentation, and I really welcome you to the BioMedics family. Please apply for the position. It's great fun to work at BioMedics. Thank you.
Great, thank you very much, Thomas, for this excellent introduction to BioMedics. I second your last sentiment there. And now we are going to hear from Wei, who will give us more information on the science behind the call. So over to you, Wei.
Thank you, Rita. So let me just share my slides, and can you see it?
Not yet.
They're coming now?
Yes, something's happening.
Okay, wonderful.
Yes.
Okay, so you can see my mouse, right? So yeah, hello everyone. It's a pleasure to be here. After listening to Thomas' talk, I'm already regretting that I've not joined BioMedics before, because I will also explain in a minute why I was in particular struggling to decide over industry or academia, and how my career path yeah, would be looking like in the future when I was a PhD student. So before we dive into the science of this call, which is enhancing tumor immunogenicity, I would like to give a brief introduction of who we are as a company, and also who we are as a research organization, also we as people, so that you understand the context of this challenge, and also how you may interact with us, what kind of career path would be benefiting from this challenge, and so on. So Merck as a company, in essence, is described by this one slogan: science is at the heart of everything we do. And I think due to the different business sectors, we have different ways how to interpret the slogan. But I think for me in particular, important is that we embrace curiosity and really try to get into the depth of science, and also try to really enjoy the beautiful part of science. But it's not only about enjoying something really, really beautiful, it's also turning this enjoyment and this beauty into really something valuable and making an impact for both the patients and also our society. So we as Merck understand us as a global company. We have the naming rights for Merck across the globe, except for North America. So in the United States and Canada, we are known as EMD Serono, but in the other corners of the world, we are known as Merck. We have over 60,000 employees, which work in over 66 different countries, and across the 3 different business sectors. So Merck is a very seasoned company with a lot of history. It's the oldest pharmaceutical company. So we recently, so 5 years ago, we actually celebrated our 350th anniversary. And 2 years ago, we were hitting around 20 billion in sales. But now last year, we actually exceeded this mark and went to 22 billion and then planning to go to 25 billion in 2025. So a lot of this money the company is making is actually reinvested into R&D activities to create new products and also new value for the patients. So our three business sectors are life science, healthcare, and electronics. And I'm coming from healthcare, in particular, the research unit oncology, which is focused on the two topics, classical oncology and also immuno-oncology. However, we are also interested in other areas where we can improve the life of patients, in particular neurology, immunology, fertility, and general medicine. So if we look at the Merck pipeline, the RU oncology clinical assets are depicted in green. We can see we have two DNA damage and repair inhibitors, ATR inhibitor and also ATM inhibitor in phase 1 clinical trials. We also have our clinically approved anti-PD-L1 inhibitor, avelumab, also called Bavencio, which is approved in different indications like Merkel cell carcinoma and also bladder cancer. And then we have another checkpoint inhibitor, anti-TIGIT, and also a metabolic checkpoint, which is a dual A2A antagonist. So you can tell from the pipeline, we have a mixture of different assets. So 3 of them are tumor immunology focus and 2 of them are DNA damage repair focus. So we have a strong interest into these 2 areas, and which basically brings me to who I am. So I'm people call me Wei. So the longer name is Wei Xiaosha, but you can just call me Wei if we will ever met. And I started actually my very ordinary industry career, actually not in the industry, but in the academia. So my first exposure to industry was actually in my diploma thesis. I was working on one of the first EMA-approved immunotherapies, which was a T-cell engager, catumaxomab. And this trifunctional antibody was developed by Fresenius Biotech. And during this time, I had my first exposure to Fresenius as industry partner, and which basically prompted me to also apply later on after I finished my diploma thesis to apply for a stipend from the Ezekröner Fresenius Foundation and participate in different graduate schools at the University of Frankfurt. And I think in Frankfurt, we had a great focus on interaction with industry. So actually the Frankfurt International Research Graduate School, so also called FIRST, was very focused on having workshops with industry, industry members, and also having visits into different pharmaceutical companies. However, still during my PhD thesis, I was struggling a little bit to decide what I would do after my PhD thesis. So I actually didn't have ever the chance to have like really great exposure to the industry. So even having these short superficial glimpses into industry life, I really didn't understand what industry research was all about. And I think the beauty about the biomedics infrastructure is actually that you have basically what Thomas said, the best out of both worlds. And during my PhD, I was very fortunate to also get advice from my supervisor, who also came from industry, and he basically told me I have to join industry as quick as possible and trying to get exposure and to really decide if I want to continue my career in the industry. And so this was why I applied for Merck for a postdoc position, and luckily I was invited to an interview and then also hired. And so after my PhD thesis, I directly started my career at Merck as a postdoc and expert and also contributor. And then over the last 9 years, I gradually developed, into different scientist levels, and also developing into a manager, mentor, and also leader of projects and also groups. So right now I'm associate director. I'm responsible for 2 labs in May in the RU oncology, and basically leading different types of drug discovery activities. I think interesting maybe for some of the participants is that you never know what life gives you, but the best way to find out what you want is really to experience it. And I think for me, I never had actually the chance to understand the beauty of industry research. So for me, it was just jumping into the cold water and experience it. But I think for you have a chance to really try to find out for yourself and also do deep self-reflection what you really want for your career and if you want to proceed in academia or in industry. So in Merck Healthcare, we have the research unit oncology, and we have different major topics. Now, I'm coming from tumor immunology department. I'm always working with trying to activate the immune system to fight the tumor. And then Julia is coming from the DNA damage response innovation cluster, and also we have interest in oncogenic signaling and antibody drug conjugates. So, and this is where we are basically heading to. So tumor immunogenicity is actually the interaction interface between the 2 innovation clusters, tumor immunology and also DNA damage and response. And luckily, I had the fortune to team up with Julia from the DDR innovation cluster, because obviously you cannot know everything, and it's a great, kind of teamwork to really get the core expertise of both of us and then combine it in an area where actually none of us has embarked to ever before. And this is why we believe that by leveraging the both expertises, we can really get the best traction. And potentially for your proposal, you may be very strong in one or the other area, and you might also need to team up with another person who might be complementary to your strengths. So the biomedics challenge in wording is to identify novel DDR targets. So targets which are involved in the DNA damage and repair biology, and we hope to improve the tumor immunogenicity through modulating those DDR targets. And hopefully we can trigger new Merck drug discovery activities with that, because if we look at the whole drug discovery value chain, so this is just everything in a nutshell. The first fundamental process is to find a target, right? And then trying to target this target by finding the right molecules, and the more many molecules, select the best molecule. And then to some extent, later on, we'll try to translate this molecule to the clinic, so preparing clinical trial design, phase 1 dose escalation, and so on, by finding the right biomarkers. And then during clinical development, we will try to verify the right dose of the drug, find the safety margin, and also the activity of the drug, and ideally demonstrate the therapeutic window, which can lead then to the approval of the drug. So in this whole value chain, you can see it's a very, very long road leading to approved drug, but the most important and probably fundamental process to actually find the right target, because if you don't have the right target, obviously the whole chain is useless. So this is where the applicants come in. Ideally, you have an idea how to set up the tumor models or set up the experimental strategy to really pursue target finding. And so this is the perfect moment to dive into the science of this whole challenge. So if we are thinking about DNA damage and repair, I think the most prominent example is PARP inhibition. And in this context, if we inhibit PARP, especially in a ovarian cancer setting, there's some clinical breakthrough here. We can see that in normal cells, actually, cells can have different kinds of DNA repair mechanisms which compensate for the loss of DNA repair by having redundant mechanisms, and this basically leads to that the DNA damage induced can be repaired and leading to cell survival. However, if we have a genetic vulnerability, so if we have in this case BRCA1 mutation, the cell actually loses the ability to efficiently repair the DNA damage inflicted by PARP inhibition, and this is leading to cell death. So this concept we actually call synthetic lethality, where we are leveraging the genetic vulnerability of the tumor and exploiting the deficiencies in certain DNA repair mechanisms, inhibit another one, and then inducing cell death. So with this concept in mind, I think those of you who are familiar with the DDR biology are pretty confident that this is the ultimate truth. However, if we look at the problem from another angle, so if we are applying a PARP targeting drug in immune competent mouse tumor models, so in vivo models, we can actually see that in this BRCA1 deficient engineered mouse model, we have a pretty good tumor growth inhibition by PARP targeting. However, the intriguing part here in this paper is actually that if we deplete CD8 T cells, which are the cytotoxic T cells responsible for attacking the tumor cell, we can see that this PARP inhibition efficacy actually goes away to some extent. So meaning that PARP inhibition actually also kind of requires the involvement of the immune system, and especially requires anti-tumor immunity to take full effect. So what is this mechanism about? So if we look into more detail into what the authors published, we can see that there are certain immune cells, so innate immune cells besides T cells, which are key playmakers of this process. So by PARP inhibition, we can see upregulation of TBK1 and RF. So these are two key pathways downstream of STING signaling. And STING is a major DNA sensor and key to inducing productive type 1 interferon response, leading to a very efficient anti-tumor immunity. And this is also what has been seen after PARP inhibition. There's an induction of type 1 interferon and also downstream T-cell chemokines, leading to basically the whole cascade of anti-tumor immunity. So what we can tell from the BRCA1 model here is that if we have PARP modulation, we have on one side the synthetic lethality concept, that tumor cells go into cell death because they are unable and incompetent to repair the inflicted DNA damage. But on the other side, we also have induction of double-stranded DNA due to this potential cell death or cell stress, and this may also lead to activation of the innate immune system, so in particular STING in this example, and then downstream type 1 interferon and T-cell activation, which further helps the immune system to attack the tumor. So this is just one example how the challenge can be interpreted. So we have a genetic vulnerability, and then we have a pharmacological inhibition, which basically leads to an anti-tumor phenotype. And so the question could be if we can find other DDR mechanisms which also help to unleash the power of anti-tumor immunity, in particular by activating the STING pathway or type 1 interferon responses. Now, this was just one example. I would like to provide you another example how we can leverage a tumor mutations or tumor genetics to enforce immunogenic phenotypes. So Some of you might know there's such a thing like tumor mutational burden, also called TMB, and this tumor mutational burden has been actually used as a biomarker to predict response to checkpoint inhibitors or anti-PD-1 antibody, which is an immunotherapy. And I think counterintuitive for classical oncology textbook knowledge is that usually you would anticipate that if you have a tumor mutation, these tumor mutations are actually driving tumor progression, right? So you have loss of function of tumor suppressors or gain of function of oncogenic drivers. However, tumor mutation can also mean that the cell, which has a lot of mutation, is prone to generating a lot of proteins which are very foreign and can be recognized in a very efficient way by the immune system. And this is actually how this works. So you can see that with the generic cutoff for TMB over 10, which is just a metric of SNV mutations per megabase. We can actually see that a lot of indications where we have approved immunotherapy actually have a very high TMB, and this is basically the concept that with this generic cutoff, you can increase the chance that actually the tumor is very immunogenic and then responding to anti-PD-1 therapy. However, if we look at the left side of the spectrum, we can see that actually a lot of tumors are not very immunogenic, and the question is, of course, for us, can we artificially increase this in some settings by pharmacological inhibition or exploiting the genetic makeup and vulnerability of the tumor type. However, I think in general, there's a lot of debate over if this generic cutoff is right or wrong, or if this is the ultimate truth for all indications. But I think it tells you a story, so that basically the tumor mutational landscape of a tumor actually defines its immunogenicity. And this is also true for another example where we have the POL-E proofreading deficiency, which leads to actually to very TMB high tumor settings where the tumor has a lot of mutation. And if we look at the swimmer plot of overall survival of patients receiving anti-PD-1 therapy, we can actually see that the survival of the patients are the highest, which have a POL-E mutation affecting TMB. There are also non-functional mutations, but that's another story. So in short, if we look at the POL-E mutation, of course, what comes into your mind is that, okay, we can leverage this genetic vulnerability to really have great anti-PD-1 responses. However, if we look at the rate of POL-E mutations, we can see that actually only a very small numbers of patients have a POL-E defect, and which means that only very small numbers of patients have a predictive genotype for anti-PD-1 therapy. However, there are also other biomarkers. So one marker is basically the mismatch repair deficiency phenotype. So during replication stress, we have a lot of replication errors, and if the tumor has a disability to repair mismatches, it's basically accumulating mutations and then leading to increase of tumor mutational burden, but also a tumor index, which can again lead to very immunogenic peptide generation. And I think one striking example is that if you pre-select patients by genetic mismatch repair deficiency, we can actually see that in this case, 2 patients have very visible rectal tumors by endoscopy, and then after PD-1 treatment, with dostarlimab, there's a very robust response leading actually to the disappearance of these tumors. So very striking data, and same is true actually for colorectal cancer, where mismatch repair deficient tumors actually benefit. So patients benefit highly from pembrolizumab anti-PD-1 therapy as compared to chemotherapy. So the bottom line of the story is actually that what I want to tell you is that mismatch repair deficient colorectal cancers, they're very prone to immunotherapy, especially to PD-1 therapy. And we can see that in this spider plot that patients are responding very robustly with this phenotype. However, if you have patients which have a mismatch repair proficient colorectal cancer, actually the response is very poor as compared to the mismatch repair deficient ones. And the logic is actually very simple here. So mismatch repair proficient tumors, you have the ability to repair their tumor mutations, and this leads to actually a low mutational rate, a low ratio of new antigens. So most of their antigens are actually self antigens, and whatever is looking like a self antigen, that's where the immune system comes in and actually generates tolerance again. So the immune system is basically breaking its activity. And there's also, as a consequence, a low ratio of immune infiltrates. However, if you have a mismatch repair deficient tumor, there's a high mutational rate, high neoantigen repertoire, so a lot of antigens to be recognized by the immune system, and then also, not surprisingly, high immune reactivity and high ratios of different immune infiltrates. So this is a very different example as the first one with the STING and type 1 interferon biology, but I think it's another example how you can tackle the challenge. So in this case, the ultimate question could be look like that we want to modulate DNA repair to really increase the tumor mutational burden, or at least a mutational signature, which can be very immunogenic and then leading to very neoantigen-rich tumors and robust immune activity after checkpoint blockade. So as a summary, I think many different ways lead to Rome. So our goal is enhancing tumor immunogenicity, and the starting point definitely is leveraging DDR defects. So defects in DNA damage and repair mechanisms, and leveraging genetic vulnerabilities to reach this goal. And I've described two examples. So you may take this as a blueprint or inspiration, or even completely ignore it. However, this is our starting point, and you can reach the final goal by different means. So by increasing tumor mutational burden, index, induction of new antigens, or generating genetic instability, and then subsequently STING signaling and type 1 interferon responses. So I will not go into details of this right part. So experimentally, I think you can line out the strategy, outline the strategy, how to do that in the actual application. So Sven actually gave me this slides. So you may wonder what kind of talents we are in particular looking for, what's the particular profile we have a strong interest into. So I think in general, I think being open-minded to cross-functional working is a big plus, and ability to either collaborate or lead a cross-functional team is certainly something which we can see as a key expertise, because I think nobody is perfect, nobody knows everything. We need to actually leverage the expertises of other people to really efficiently break into innovative areas where nobody has ever been before. And for the group leader positions, we're actually in particular looking into someone who has relevant postdoc experience and ability to also act as a mentor and guide a team of more less experienced postdocs. So these are our industry mentors: Julia, my partner in crime in DDR research, and also Sven, who is a senior innovation manager from strategic innovation, supporting us in the operative questions. So with this, I'm handing back to Rita or Thomas, and looking forward to the Q&A session. Thank you for your attention.